Hi, we're here to talk about GMOs. See all these interesting things on the table? That's to help you to tell me something. What do you know about GMOs? Have a look around here. What do you think this is showing us? What do you know about this? Are we eating GMOs?
Yes.
Yep. Yeah, we are eating GMOs. Every day you're eating GMOs. Which of these things do you think have got GMOs in them? Yeah?
The flour, maize.
Mealie meal. Yes, South Africa grows genetically modified maize. So we've got, that's GMO. So, will it be in cornflakes? Yes. Now, what about baby food? This is always quite an emotive one. Is there GMO in baby food? What do you think?
Yes.
I think so.
Maybe.
Yes. Yes, some foods there are. I mean, some makes will say they're organic and will actually state on the bottle, no GMOs. Others won't say whether they've got GMO in them or not. If there's soy in it, and there's often soy from soya plants, soy protein, then most of the soy protein that we import into South Africa or we grow here is also genetically modified. What about my made in South Africa cotton t-shirt? Do you think that's GM cotton?
Yes.
It is actually, yeah. We'd grow GM cotton in South Africa as well. So why? Why do we do it? Is it good or is it bad? What do you think about that? We're going to find out a bit more then. Maybe by the end of the workshop we'll know a little bit more about it. Let's have a look at this fruit. Who wants any of these delicious pieces of fruit? Yuck, all right. Is this more the sort of thing we're thinking about?
Yes. Yeah.
Yes. Now, do you think it's possible to genetically modify things so that, in fact, they don't ripen on the shelves until we're ready for them?
Yes.
Yeah, that's one of the ideas of genetically modifying organisms, to do that. What have I got here? A square
banana.
A square banana. Now, this is one of the criticisms of GM from the consumers. What, would they want a squared banana? Do they care? Who might find a square banana advantageous? Think of packaging, right? Think of the boxes that the fruit goes into. If you've got square bananas, you can just stack them up in the boxes. Imagine, rectangular watermelons, how much easier it would be. Such things are theoretically possible. There's another good use for bananas that has actually advanced quite far so far, and that is to make vaccines, to make the bananas make vaccines. So introduce into the banana plant a gene that will be able to make something that will serve as a vaccine. For example, for measles. So instead of the sister having come round and inoculated all the kids in the country just recently, each school will be sent a few box loads of bananas. Kids will eat the bananas and get their vaccines that way. So the vaccines won't have to be stored and refrigerated. That's quite a good idea. Here I've got some rice, and rice is the staple food in many parts of the world in Asia, and so scientists have actually made a genetically modified golden rice that contains a precursor of, makes carotene actually, which is a precursor of vitamin A, so that the kids won't suffer possibly from complications like blindness. So genetically modified organisms can do a lot of good. They also raise some concerns. What's new about it all? What's special about genetic modification? You see, for centuries, for thousands of years, farmers have been breeding plants and animals to get ones that give bigger, better crops, more milk, sweeter apples, bigger ears of corn, and so on. The thing about genetic modification is that we can use the whole new DNA technology, and we can introduce a gene from one species and put it into another species. So you can get a combination that would never occur in nature. Look, we've got white flowers here, and these flowers will never make blue flowers because they don't have the gene in them that will make a blue pigment. But think of a blue flower, like a delphinium. You can take the gene that makes the blue color for delphinium and put it into these plants, and then you can grow blue plants, which are going to have resale value. So really, the sky's the limit in what one can think of. Look, beautiful, fresh, red, shiny tomato. At one stage, scientists thought, why don't we put a gene in here from a fish that can swim under the ice and doesn't freeze? It's got its own kind of antifreeze in it. it has a gene that makes a compound that stops the fish's blood freezing. So the idea was, oh, let's put that gene into a tomato plant, and then the tomatoes won't wilt at the first frost. What do you think people's concerns were there? Would that plant taste of fish? Would it? No, it's just the one gene that makes that one compound. Would vegetarians say, oh, we can't eat this, it's got an animal product in it? Yes, they possibly could. That experiment actually didn't work very successfully, so it didn't go to market. But lots of other things have worked. Look at these plants. Say they were a crop plant. Say this was a mealy leaf. What's the problem here? Yeah, so what's been happening to it? Yeah, insects are eating it. So as you know, crops are sprayed with insecticide. Here's my Wonder Garden gun. If it's a whole field, then the crop sprayer has to come and spray all over the field. You know about the disadvantages with that. So genetic modification allows scientists, and this already happens, to take a gene that kills insects and put that gene into a mealy plant, a maize plant, for example. So any insect that eats that plant will get killed by the built-in insecticide. Then the crops don't have to be sprayed. Do you think it's a problem then that you're going to eat that mealy plant? What's going to happen? Are you an insect? No. The toxin, the poison that kills the insect in this particular case is totally safe for humans and for the cows that eat the remains of the mealy. So that's good. Another thing that's been introduced into crop plants is a herbicide, a weed killer. And again, the gene's been put into those plants, so those plants can grow in the presence of a herbicide, a weed killer. The fields can then be sprayed with lower levels of herbicide than would be used normally. These are the things that are put forward as the good, and then there are things that are the concerns. And we can have a look and discuss that a little bit later. I think the next thing we need to talk about is how is a genetically modified organism made then? You've got on the table the flow diagram, and I'm going to talk us through the flow diagram for how to make a genetically modified plant. And I'm going to ask you just now to design your own genetically modified plant. So you can already be thinking about that. What sort of desirable characteristic, one calls it a trait, would you like to introduce into something? Look, we got some innocent goldfish lying there. Actually, genetic modification can happen in animals as well, and they've introduced a gene that makes a fluorescent protein into some species of fish for tanks and goldfish bowls, and so those fish will fluoresce in certain lights. So you can imagine that's a must-have for somebody who's got everything else. You can now have fluorescent fish, for example. But let's talk about something that is useful, that a lot of effort's being put into this. Let's pretend, here's our maize plant. Now, this maize plant needs to grow under certain conditions, and it actually needs quite a lot of moisture, as you hear every year, the big problems because the rains have failed. So what about if we could introduce a gene into it that would allow it to grow in more arid conditions, like our cactus here? And when you look at the flow chart you've got there, you'll see we've got a side that I call the donor plant. That is the cactus that you can see in your drawing. And we want to introduce the gene from the cactus and get it into our acceptor plant, the plant that we're going to modify. So the mealy will be able to grow in more arid areas, so the farmers will be able to farm on what's called marginal land, and we'll be able to produce better crops and have less disasters than we have with food. So that is one of the ideas. And I'm just going to talk you through how a genetically modified organism is made before you design your own for me. It's quite a complicated process. And the first thing we do is identify our donor plant and extract DNA from it. The DNA is extracted, one has the strings of DNA. And this is going to represent a string of plant DNA. And on it, these beads represent the genes. And there's maybe 30,000 or 40,000 different genes in the genome. That means the total content of DNA, all the chromosomes. Imagine all the chromosomes are here. And we need to know which gene on here it is that will confer drought resistance to our maize plant. The way we know which gene to use to take out is actually the science of bioinformatics. And I'm going to talk about bioinformatics later. But there are ways to identify the genes here. And let's say that that science tells us that the gene we need is this little orange gene that you can see in the middle here. And we need to cut that gene out of the DNA and put it into the mealy plant. Sounds easy, doesn't it? It's black box technology. Right, so the way this gene gets cut out is by using molecular scissors. They're called restriction enzymes. They're enzymes that exist in some bacteria and which scientists use. So this is really high-tech biotechnology. And scientists use the restriction enzyme, represented by these scissors here, to cut the DNA strand to release the gene that I want. And the rest of it, I don't need. This is the gene that I've got to put into the maize plant. Now you can imagine, I can't just throw it at a maize plant. How am I going to get it into a maize plant? So now quite a lot of manipulation has to be done. Biotechnology, plant molecular biology. One of the things the scientists do is make lots and lots of copies of this gene, first of all, using a process called PCR, polymerase chain reaction. And they're doing all this in those little tubes, like I showed you before, in the laboratory. And so what they would have in the end is maybe 1 million molecules in their little tube. I'm just representing 2 of them here. So we made lots and lots of copies of the gene that we want to introduce into the maize plant. So if you're following me down here, you'll see we've chosen our plant, we've got out the DNA, we've cut out our chosen gene, which is the orange gene here. Now, we can't just throw it into the maize plant. We've got to introduce it into something we call a vehicle. The name for it is a vector that allows it to be carried, because vector meaning carrier, allow it to be carried into the maize plant. And the vectors we use, I'm representing here in this little bucket, but it would be in those tiny little tubes. And these are also DNA. And these are circles of DNA, which have actually originally come from bacteria. And again, biotechnology has allowed us to use them in the laboratory. The nice thing about DNA is DNA is DNA is DNA. Whether it comes from a bacterium or a plant or an animal, it's that same basic structure. And so I can use these plasmids as the vehicle to introduce the DNA into a maize plant. Scientists have come up with a few other little tricks as well. So they'll have thousands of these little plasmids, and they have already engineered the plasmids to look something like this. They've introduced some more genes onto the plasmids. This represents a gene, and this represents a start and a stop signal. So this gene will get read in the plant it's going to be put into. That's this part of your picture. This represents it linearized, cut open like that. So there's the chosen gene, and there's its start signal and its stop signal. Now, what we have to do is join the gene we want to introduce into the plant with this. And that's also all part of molecular biology. It's quite clever, because this has little sticky ends that will attach to this. And so I represent it by tying it up like that. And the other end, it's actually an enzyme called a ligase that joins them together like this. And so now we've got this structural circle structure. And it's got the gene we want, and it's got a special marker gene that will allow us to recognize the maize plant that has got this in it. So this is what I'm representing here. I'm calling it a DNA cassette. And now this is ready to be put into the maize plant. How? How do we do that? That's the second step of this process, and this is what I'm talking about on the other side here. This is a process known as plant tissue culture, also biotechnology. If one takes the growing tip of any young plant and pulls the growing tip out, and puts it into a Petri plate, which I'm representing by this little container here, in the laboratory, and gives it the right nutrients and puts it in the right light and the right temperature, the cells at the damaged tip divide and divide and divide to form something we call a callus, which is to plug the wound, basically. It's like a blood clot, but for a plant, and it's cells. And I'm going to represent it in this dish by this little green blob, which is Play-Doh, but think about it as being thousands and thousands of plant cells. And now what we have to do is shoot, and that's really how it happens, shoot this into our plant cells. So we will have taken a growing tip of a nice young fresh maize plant, not one that looks like this, and put it into a dish with the right nutrients and let it grow this mass of cells. And now we shoot into this mass of cells our recombinant molecule, ultimate high-tech biotech. And I'm going to represent it by using this gun here. If you look on your chart, you'll see a gene gun is used. It's quite a sophisticated piece of apparatus, of course. But for the sake of our simulated experiment here, I'll attach my recombinant molecule to my gene gun. Here's my callus tissue. In real life, this would be put into this fancy machine, and then the fancy machine bombards the callus with hundreds and hundreds of these little recombinant DNA molecules with this gene in them as well. So here we go. For my simulation, I'm going to shoot my recombinant molecule into there we go. And it's shot the DNA that I want, the gene that I want to put in there. It shot it, more than one, thousands of them will all have bombarded it, and several of them will have gone in. And then I take the whole mass of tissue and put it into another little Petri plate. I add nutrients, the right nutrients to it. I add nutrients that encourage shoot and root growth, plant hormones. And in a certain period of time, what will happen is little green plantlets, these are meant to represent maize plantlets, will grow up. And you see the little orange bead in them? That's representing the gene. And every cell will have a gene inside it. Now, obviously, I can't represent it here. So this plant has now got the gene in it that we've taken from cactus, and it will confer, we hope, a resistance to drought. The marker gene that I mentioned will have just been used as part of the experiment and part of the manipulation to make my chances of this happening higher. Once I've got the little plantlets like that, I can pick them out with tweezers under sterile conditions. So we'll be doing that. So I'll pick one out. I need one with the gene in it. Pick it out and plant it in little jars like this. Put them in the light at the right temperature in the greenhouse. And I'll put each plantlet in its own little jar. And then if my experiment is successful, I will have all these little jars, and these are clones as well, because they're all the same genetically as each other, and they will be grown up in the greenhouse and subsequently planted out in the field for field trials. And if we're lucky, we're going to have this new mealy plant that's going to grow very happily in the Kalahari Desert. So that's how genetically modified organisms are made. Now, what I want you to do is design your own plant together. Introduce some characteristic into a crop plant, or anything you like, actually, that you think will be marketable. So now I need you to discuss some ideas. Right.
Yes, son, I bought you some new spinach.
You hate spinach.
Oh, but don't worry, it's strawberry flavour.
Don't lie.
No, try.
Wonder how that happened.
Well, what the scientists did was take the gene that gives us the strawberry flavor and put it into the spinach plant, which then gives the spinach a strawberry flavor. Hence, the spinach was genetically modified to taste like a strawberry.
Very nice.